guys, welcome back to another episode of Cycle of 3. Now today, we're going to talk about the new invention, PCR, also known as polymerase chain reaction. For this episode, we got one special guest, Carrie Mullis. Hey Carrie, can you tell me more about yourself?
I was born on December 28, 1944 in North Carolina. I took college at the University of Carolina and majored in chemistry. Then I earned a degree. I also majored in biochemistry and then a PhD in biochemistry. I also made 3 books. My most popular book was Dancing Naked in a Minefield. It's about my history and how I made polymerase chain reaction.
You know what's cool? I also went to North Carolina.
Oh, really?
Yeah, I majored in chemistry, and I'm also one of the professors in Harvard. Now I'm in a podcast talking about science. I heard PCR is used for scientists to collect small samples of DNA to do important experiments. Could you go more in depth for me?
Yes, you are right. It's used to collect samples of DNA. It can also be used for crimes, understand disease, and developing new treatments. We found out it can be used to find out if you have cancer, genetic mutations, and identifying pathogens.
I'm going to take a guess on how it works. Is it like heating and cooling operation which separates the DNA?
You are correct, but there are more operations to it. Like, we have to get the machine going.
Sorry to interrupt, but how much did it cost to create the machine?
So, we have to do like a lot of tests, and it kept failing. So, probably around $100,000 today, correct?
Oh! My, that's a lot. Back to what you were saying on PCR, how it works, mind explaining?
So basically, it goes through 3 processes. The first thing is denaturation. Denaturation is basically when the DNA that is needed to separate its 2 things. The second process is annealing. Annealing is when the temperature gets lower to the primers, which are short DNA sequences, and can attach to the single strands of DNA. The final process is extension. Basically, when DNA polymerase enzymes add nucleotides to build new DNA strands, these processes is repeated many times, resulting in numerous copies of specific DNA sequences.
Dang, that's a lot, kind of. But I thought it'd be more simple since you're a good scientist and all. Carrie Mullis, I know that what you said was a summary, but the viewers are probably curious. You mind going more into what you were saying?
Yes, so during denaturation, heating the DNA to around 95 Celsius is crucial because it breaks the hydrogen bond between the base pairs, eventually separating the double helix into 2 single strands. This extreme heat is why we often use a special type of DNA, polymers, like Taq polymerase, which is heat stable and won't denture itself. For annealing, the temperature is lowered between 50 to 66 Celsius. This allows the single-stranded DNA polymers to bind specifically to their sequence of each of the separated DNA strands. These polymers are absolutely crucial because they provide a starting point for the DNA polymers, as the enzymes can only add nucleotides to an additional 3 hydrogen group. They also define the specific region of DNA that will be amplified. Finally, in the extension phase, strands by finally at the extension at about 72 Celsius, the DNA polymers, like Taq, as I said in denaturation, synthesizes new DNA strands by adding exonucleotides in the 5 to 3 direction, using a single-stranded DNA as a template and a new primer as a starting point. This results in the doubling of the target DNA sequence in each cycle.
It's so smart. Repeating these cycles many times leads to an exponential amplification. Theoretically, if you start with only a copy after 30 cycles, you'd have over 1 billion copies, right?
Exactly. That is correct.
I also have a quick question. What type of information does this provide about cells and the process?
Well, Professor, what PCR gives us is a window into a specific blueprint of life at a resolution previously impossible without cloning. Before PCR, if you want to look at a particular gene or even a small sample of DNA in a cell, it was like trying to find a specific grain of sand in the desert. You have to clone it. You go to bacteria, hope you got the right place. It's a real pain in the neck. It took weeks, even months.
Basically, what you're saying is what PCR does is magnifying that specific grain of sand by making billions of identical copies virtually overnight?
Yes, exactly that, Professor. Overnight, we can make a billion of copies. Any other questions?
What was the biggest problem you faced getting PCR to actually work in the lab? Was finding a heat-stable polymerase for the first solution, or did you try the other options first?
The main problem was the DNA building enzyme itself. To start PCR process, we have to heat the DNA to have a very high temperature that separates two strands. The enzymes we were using, however, couldn't keep up with that extra heat. They would denture or just break down after each heating cycle. This means after every single round of heating, we had to manually add fresh enzymes, as you imagine. It took a lot of time, a lot of money, and limited on how many DNA copies we could make. At the moment, we discovered tap polymers. This enzyme is sourced from bacteria found in hot springs, meaning its natural heat is stable. It could endure the repeated heating and cooling cycles of PCR without breaking down, eventually building a new DNA strand every time.
Would you sell this like your company when you pass away?
No, I wouldn't be selling anything actually. My legacy, if you want to call it, that isn't about money or ownership after I die. It's about the fact it works, and it's used every single day in every lab around the world, and making discoveries, diagnosing disease, and capturing criminals. That just means a lot to me.
For our listeners that might not be the best at science, why should they care about the technique like PCR in their everyday lives?
Well, I think the viewers watching right now, they should care, because what PCR does is, if you ever gotten sick, in that case, it identifies and does tests to see what you have. For example, COVID or the flu. What it also does is, if you ever have watched those detective movies like Dexter, what it basically does is they collect blood or hair and identifies who the person was. That's PCR for you.
Well, there you have it, guys. That's why you should probably consider PCR in your life, polymerase chain reaction. As we wrap up today's episode with our special guest, Carrie, and the topic polymerase chain reaction, I want to ask, what do you believe is the single most important in this contribution to science or society?
Honestly, the biggest contribution of PCR is that it lets us to take tiny bits of DNA and make them big enough to study. That simple idea opened the door to modern genetics, better diagnostics, and countless discoveries. It's basically gave us scientists a new way to see life in its smallest scale.
Well, all right, guys, that is the end of the Cycle of 3 podcast. Be sure to stay tuned for more uploads in the future, more podcasts. This is our ending. We all say goodbye to Carrie. Bye, Carrie.
Bye.
Remember, like, subscribe, hit that bell. See you next time.